Prophase 1 starts off with chromosomes condensing, then the nuclear envelope starts to break down and spindles form. Centrosomes move to the opposite ends, and microtubules attach to kinetochores. Homologous pairs connect during synapsis. Here they cross over. Crossing over is the exchanging of segments of DNA between non-sister chromatids, which forms the chiasmata.
Homologous chromosomes line up at the metaphase plate. Then, chromatids attach to microtubules coming from both of the poles. Homologs separate and are pulled to opposite poles. The sister chromatids are still attached at this point.
In telophase 1 and cytokinesis, there is a haploid set of duplicated chromosomes on each half, and then a cleavage furrow or cell plate develops and the cytoplasm is split into two.
Prophase 2 is the similar process to prophase 1. The nuclear membrane will start to break down and then chromosomes will condense. And centrosomes will appear and go to the opposite ends of the pole, and then the spindle fibers will connect to the kinetochores of the chromosomes. In metaphase 2, a metaphase plate is created and the centrosomes will go on the opposite sides and chromosomes will line up at the metaphase plate. In anaphase 2, the sister chromatids are pulled apart by the spindle fibers and they're pulled to opposite sides of the cells. In telophase 2 and cytokinesis, a cleavage furrow begins to appear, and the chromosomes are on opposite sides of the cell, and the nuclear membrane will start to reappear around the chromosomes, creating 4 unidentical daughter cells. Sources of genetic variation. Crossing over occurs in prophase 1. It is the recombination of linked genes on homologous chromosomes, and it's the greatest source of genetic variation. The law of independent assortment says that different alleles will sort independently of each other. There are 1 out of 8.4 million possible chromosome combinations, and random fertilization is when one random sperm penetrates an egg. There are 70 trillion diploid combinations that can come from this.
Some errors in meiosis include non-disjunction. This is when sister chromatids fail to separate during anaphase, causing uneven distribution of chromosomes in daughter cells.
Some errors that occur during meiosis are trisomy 21, which is also known as Down syndrome. Another would be Klinefelter syndrome, in which a male has an extra X chromosome, and you would display the male phenotype but have secondary sex characteristics of females. There's also Turner syndrome, in which a female only has 1 X chromosome, in which a female development is altered, and it may lead to things such as infertility. All of these disorders are because of non-disjunction.
Without meiosis, genetic variation wouldn't exist, and that's necessary to sustain a population. If scientists could figure out how to ensure that checkpoints in the cell cycle are properly working, then disorders such as Turner and Down syndrome wouldn't be prevalent. Be sure to pay attention to details. It's not stressed on the AP exam, so don't spend hours trying to figure it out, and enjoy the class and have fun because the year goes by quickly.